Hello and welcome everybody to my channel Omini Bioentrance, and today we will be discussing Karnataka State Eligibility Test Life Science Question Paper 2 conducted in the year 2020. So coming to the first question. The constituent monosaccharide in chitin. So chitin is an acetylated polysaccharide composed of N-acetyl D-glucosamine groups linked by beta 1,4 linkages. So answer here is option C, that is N-acetyl D-glucosamine. An example for aromatic amino acid is, so I have drawn the structure of each of the amino acid, and you can see that tyrosine is the one which is having an aromatic ring, and hence it is an aromatic amino acid, whereas methionine, alanine and valine are not. So the answer here is tyrosine. Then next one. Which functional group is never found in alpha amino acids? So alpha amino acids are simple molecules that are made of a central carbon called as alpha carbon that is bound to primary amine group, that is amino group over here, and a carboxylate group over here, and to a side chain R. So it doesn't have CHO. So the answer is option C. Whereas SCH3, that is option D, it is an amino acid side group. So answer is option C. Then next one. Ramachandran plot displays. It displays allowed angles of phi and psi for polypeptide backbone. Answer is option A. Then next one. pH is equal to pKa when. So here you have got the pH is equal to pKa plus log salt by acid, that is Henderson-Hasselbalch equation. They are asking when pH will be equal to pKa. So salt has to be equal to acid. So if we take concentration of salt equal to concentration of acid, you get it as log 1, and log 1 is 0. So pH is equal to pKa, that is proton acceptor, that is salt is equal to proton donor acid. So answer is option B. Then next one, left-handed helix is found in. So it is found in Z DNA. A and B DNA are right-handed. Then next one, match the following with reference to protein structure and modifying agents. So mercaptoethanol, it is disulfide bridge 1B. Then guanidine hydrochloride is for hydrogen bond, urea is for hydrogen bond, and dithiotritol, it is for disulfide bridge. So the answer here is option B. This urea and guanidine hydrochloride, they disrupt hydrogen and hydrophobic bond. And mercaptoethanol and DTT, that is dithiotritol, it reduces the disulfide bonds. Then next one, match the following. Column A consisting of different vitamins and their names in column B. So vitamin D, it is calciferol. Vitamin E, it is tocopherol. Vitamin B6, it is pyridoxine, and vitamin B1, it is thiamine. So answer is option C. Next one. Where are the ion carriers located in a cell? So plasma membrane, it regulates the flow of ions across it, and the flow of ions occurs through special ion carriers and channel proteins present in the plasma membrane. So the answer here is option A, that is cell membrane. Then which of the following cell organelles contain DNA? So it is present in case of mitochondria, that is extra chromosomal DNA. Answer is option D. Then in which of the following organelles beta oxidation of long chain fatty acid is initiated with an object of shortening the chain length? So peroxisome are responsible for beta oxidation of very long chain, that is greater than 20 fatty acids, dicarboxylic acids, 2-methyl branched fatty acid, prostaglandins, leukotrienes, and the carboxyl side chains of certain xenobiotics and bile acid intermediates, di and trihydroxycoprostanic acids. Whereas mitochondria, it oxidizes mainly C16 to C20 chain fatty acid. So they have asked in which of the following organelles beta oxidation of long chain fatty acid is initiated. So it is in the peroxisome. So answer is option B. Then next one. Cell division which consists of nuclear division, that is mitosis, followed by cytoplasmic division, that is cytokinesis, it occurs during the M phase. Answer is option D. Which of the following pair matches with membrane transport processes and its primary function? So, exocytosis, the movement of macromolecules into the cell. Exocytosis is a form of active transport and bulk transport in which cell transport molecule out of the cell, not into the cell, it is out of the cell. Then next one, pinocytosis, the uptake of water and solutes into the cell. So B is correct. Then osmosis, it is passive diffusion of small solutes, that is also correct. Then phagocytosis, it is the secretion of large particles from the cell. Phagocytosis is the process by which cell uses its plasma membrane to engulf large peptides, giving rise to an internal compartment called as the phagosome. It is one type of endocytosis. So both B and C are correct over here. Then the core of the nucleosome consists of. So the nucleosome consists of approximately 146 or 147 base pair of DNA wrapped around a histone octamer consisting of 2 copies each of H2A, H2B, H3 and H4. So answer here is option B. Then next one. The concept of pH homeostasis is. So it is the ability of the microbe to control its internal pH. Answer is option A. Then next one. A metabolic pathway that involves part of the Krebs cycle plus 2 unique enzymes, malate synthetase and isocitrate lyase is. So glyoxylate cycle, it is a variation of the tricarboxylic acid cycle, is a metabolic pathway occurring in plants, bacteria, protists and fungi. And this glyoxylate cycle, it centers on the conversion of acetyl-CoA to succinate for the synthesis of carbohydrates. And the key enzymes that are involved in the glyoxylate cycle are malate, synthetase and isocitrate lyase. So the answer here is option A, that is glyoxylate cycle. Then next one, the maximum coiling of chromosomes is observed during. It is observed during the metaphase of mitosis. Answer is option B. Then next one, during replication, the long strands of DNA in a cell nucleus get tangled, making it harder for a cell to read genes, but dash enzymes can prevent tangling. So DNA replication, these long strand of the DNA in a cell, they get tangled, making it harder for the cell to undergo replication or to read the genes and result in synthesis of the new DNA strand. So which enzymes they prevent tangling? The tangling here means supercoiling. So it is DNA topoisomerase that is involved. Answer is option C. Then which of the following are non-coding RNAs? So it is option D, that is rRNA, mRNA and tRNA. They are all non-coding RNAs. Then next one. In the final step of protein synthesis, the folding of many proteins is made more efficient by a special class of protein called. So chaperones, they are usually involved or primarily involved in protein folding. Answer is option C. The next one, which of the following processes take place in the 3' to 5' direction? So it is RNA editing. There is an editosome that can edit only in the 3' to 5' direction along the primary RNA transcript. So it is taking place in RNA editing. Answer is option A. Then next one. In eukaryotic cell, the tRNA gene, 5S rRNA gene and some snRNA genes and genes for other small RNAs are transcribed by RNA polymerase 3. That is option C. Then the role of gRNA. gRNA is guide RNA. So it is involved in RNA editing. A similar question was asked just the previous year in the same exam, Karnataka state eligibility test exam of previous year. They have asked the same question. Somewhat similar question, not the same way, but somewhat similar. Then there is, and one more thing I would like to suggest is that please go through with the RNA editing mechanism. Questions are being asked, not only for this exam, but for other exams also I have seen questions being asked regarding RNA editing. Then next one. DNA repair pathway that can repair the damage caused by large change in the structure of the DNA double helix is called. So we will see one by one. First is base excision repair, that is a single damaged base is recognized and removed from the DNA molecule. That is, if there are uracil containing bases in DNA, then uracil is occasionally and it is occasionally incorporated during the DNA synthesis stage. And this can or even the uracil can form as a result of deamination of cytosine. That is a single damaged base. Here it is uracil that arise during misincorporation during DNA synthesis or due to the result of deamination of cytosine, that is can be recognized and removed from the DNA molecule by base excision repair. Then nucleotide excision repair is the main pathway used by mammals to remove bulky DNA lesions such as those formed by UV light, that is thymine dimers or environmental mutagens and some cancer chemotherapeutic adducts from DNA. Then DNA mismatch is for recognizing and repairing erroneous insertion, deletions and misincorporation of bases that can arise during DNA replication and recombination, as well as repairing some forms of DNA damage. Then DNA interstrand crosslink repair. And these can be, that is DNA interstrand crosslinks, they can be repaired by homologous recombination or non-homologous end joining. So they have asked DNA repair pathway that can repair the damage caused by large change in the structure of the DNA double helix is called. So it is called as nucleotide base excision repair, because it is the pathway used by mammals to remove bulky DNA lesions such as those formed by UV light, then environmental mutagens, etc. So the answer here is option D, that is nucleotide excision repair. Then fermentation of one molecule of glucose to ethanol and carbon dioxide requires how many oxygen molecules? Dash oxygen molecules. So it requires 0 oxygen molecule. Fermentation is an anaerobic process. So answer is option C. Then which of the following is signaling molecules does not bind to nuclear receptors? So nuclear receptors, it is bound by steroid hormones, that is cortisol, testosterone, vitamin D3, all will bind except acetylcholine. Acetylcholine receptor is a membrane protein that bind to the neurotransmitter acetylcholine. So answer is option B. Then next one. Which of the following describes cell junction? So occulting and claudine mediate cell-cell contact. So tight junctions, these are intercellular contacts that seal the space between individual cells of an epithelial sheet so that they can collectively separate tissue compartments. And the tight junctions, they regulate passage of ions and small molecules between cells. And occulting junctions are tight junctions. And tight junctions are composed of proteins called as occultins and claudines. So which of the following describes cell-cell junction? Okay, they have asked for cell-cell junction. Then adherence junction occur as, so option A doesn't describe cell-cell junction. It is tight junctions. Then adherence junction occur as continuous bands. Adherent junctions and tight junction encircle the cell, whereas desmosomes and gap junctions are restricted to a particular site between adjacent cells. So adherence junction occur as continuous band, that is correct, and it is the next one. Occulting junctions are abundant in cells under stress. Anchoring junctions are widely distributed in animal tissues and are most abundant in tissues that are subjected to severe mechanical stress. So it is not occulting junctions, it is anchoring junctions. And inorganic ions are able to pass through anchoring junction to permit cell coupling. Gap junctions allow inorganic ion to pass through, not anchoring junction. So that is also not correct. So answer here is option B, that is adherence junction occur as continuous bands. Then next one. Gram-negative bacteria produces which of the following as their signaling molecule in quorum sensing? So quorum sensing is a regulation of gene expression in response to fluctuations in cell population density, and quorum sensing in bacteria produce and release certain chemical signal molecules called as autoinducers that increase in concentration as the cell density increases. Then gram positive and gram negative bacteria, they use quorum sensum communication circuits to regulate various activities such as symbiosis, virulence, competence, conjugation, antibiotic production, motility, sporulation, and biofilm formation. And gram-negative bacteria use acylated homoserine lactone as autoinducer, and gram-positive bacteria used processed oligopeptides to communicate. So here they have asked gram-negative bacteria produce which of the following as their signaling molecule in quorum sensing. So it is N-acyl homoserine lactones that is produced by gram-negative bacteria. So answer is option A. Then which of the following is a tumor suppressor gene? So tumor suppressor genes, they are normal genes that that slow down cell division and repair DNA mistakes, and also they will tell the cell when to undergo apoptosis. When the tumor suppressor gene is mutated, it results in the loss or reduction in the function which can lead to cancer. So here the tumor suppressor genes are retinoblastoma and P53. Both B and D are correct. Then Rao sarcoma virus was first discovered in chicken. Answer is option D. Then MHC molecules on antigen presenting cells which presents foreign peptides to helper cells is. So here class 1 MHC, class 2, class 3, etc. they have given as option. So cytotoxic T cells, they recognize foreign peptides in association with class 1 MHC molecules, whereas helper T cells, they recognize foreign peptides in association with class 2 MHC molecules. So MHC molecules on antigen presenting cells which present foreign peptides to helper T cells. So it is class 2 MHC molecule. Answer is option B. The correct sequence of events in producing an antibody response to an antigen is. So antigen presenting cell, T helper cells will activate the B cells and they differentiate into plasma cells and secreting antibody. So answer is option A. The next one, a living microbe whose virulence is destroyed and is used for vaccination is considered A. It is considered as attenuated. And what is toxoid? Toxoid is an inactivated toxin whose toxicity has been suppressed by chemical or heat treatment, but other properties, that is its immunogenicity, has been maintained. Then next one, which of the following is an autoimmune disease? So multiple sclerosis, it is an autoimmune disease, and it happens when the immune system attacks the myelin sheath. Then next one, which of the following is not the portal of entry for bacterial pathogens in the plant host? So plasmodesmata are intercellular pores connecting adjacent cells, allowing membrane and cytoplasmic continuity, and they are essential routes for intercellular trafficking, communication and signaling in plant development as well as in defense. So which of the following is not a portal of entry of bacterial pathogen into plant host is plasmodesmata. So the answer is option D. Then choose the correct match. IgA, it is a secretory component. D 3. IgE is basophil. IgG is it can cross the placenta 5, and IgM is a pentamer. So the answer here is option D, that is I3 2 1 3 2 1 3 5 4 4. Then, during morphogenesis, the formation of anterior structure in the Drosophila embryo requires the product of dash gene. So, bicoid, it is expressed in the ovary and regulates the expression of gene responsible for anterior structures, and it also stimulates hunchback. And hunchback, it is expressed in the embryo and it regulates transcription of gene responsible for anterior structures. So both A and C are correct over here. Then next one. During microsporogenesis, the tapetum is formed from. So tapetum, it is the innermost layer of the microsporangium and it provides nourishment to the developing pollen grains. So you have got the young anther and it consists of a homogeneous mass of parenchymatous cells and it is surrounded by the epidermis. Then the anther, as it grows, it becomes four-lobed, and in each of the four lobe, some hypodermal cells, they begin to act as archisporial initials, and these archisporial initials, they will divide into an outer parietal cell and an inner sporogenous cell. The parietal cell, it divides to form 3 to 5 layer, that is endothecium, middle layer and tapetum, whereas the sporogenous cell, it divides to produce a mass of sporogenous cells or microsporocytes. So during microsporogenesis, the tapetum is formed from the parietal cells. Answer is option B. Then floral development in Arabidopsis and Antirrhinum is under genetic control of dash genes. So you have got the ABCD model of flower development, which proposes the floral organ identity is defined by 5 classes of homeotic genes, that is A, B, C, D and E. And A and E class, they form the sepals, that is the first floral whorl. Then A, B, E form the petals, the second whorl, and B, C, E forms stamens, that is the third whorl, and C and E class protein complex forms carpels, that is the last or the fourth whorl. So floral development in Arabidopsis and Antirrhinum, it is under the control of homeotic genes. Answer is option B. Then which of the following is the master gene involved in sex determination in Drosophila? So it is SXL, that is sex lethal, is the master switch gene for somatic sex determination in Drosophila melanogaster. In XX animals, SXL becomes activated and imposes female development, whereas in XY animals, SXL remains inactive and male development ensures. So answer is option A, that is SXL. Then next one, products of maternal genome molecules that are placed in the Drosophila egg are. So that is RNA. If you take the example of bicoid gene, it is transcribed in the ovary of an adult female during oogenesis, and this RNA, it gets incorporated into the cytoplasm of the egg and it gets anchored to the anterior end of the egg. When the egg has been laid, bicoid mRNA is translated into bicoid protein. So product of maternal gene molecules that are placed in the Drosophila egg is as in the form of RNA. Answer is option C. Then next one. Shinya Yamanaka received his Nobel Prize for the discovery of. So it was the discovery of induced pluripotent stem cells. And these are a type of pluripotent stem cells derived from adult somatic cell that have been genetically reprogrammed to an embryonic stem cell like state. And Shinya Yamanaka lab in Japan who showed that introduction of spore-specific genes, that is MYC, OCT3x4, SOX2 and KLT4, collectively known as Yamanaka factors. They encode transcription factors that convert somatic cells into pluripotent stem cells. So answer here is option A, that is induced pluripotent stem cells. Then next one, caspases are involved in the process of. So caspases are involved in the process of apoptosis. So initiator caspases, they initiate the apoptosis signal, while the executioner caspases, they carry out the mass proteolysis that lead to apoptosis. So caspases are involved in the process of apoptosis. That is option C. Then Q cycle occurs in the mitochondria as well as in the chloroplast. Its main function is. So Q cycle, it refers to the sequential oxidation reduction of the electron carrier coenzyme Q in mitochondria and plastoquinones in the photosynthetic system. So it is coupling two electron transfer with one electron transfer. Answer is option A. Then next one, C4 plants are photosynthetically more efficient than that of C3 plants because of the absence of dash in C4 plants. So it is because of the absence of photorespiration. Answer is option C. So next question, which of the following is not a plant growth hormone? It is jasmonic acid. Jasmonic acid and its derivatives, a amino acid conjugated to jasmonic acid, amino acid conjugate of jasmonic acid, that is jasmonyl isoleucine. They are all signaling compounds involved in the regulation of cellular defense and development in plants. So the answer here is option A, that is jasmonic acid. Then next one, the fixation of carbon dioxide in C4 cycle take place by oxaloacetate and phosphoenol pyruvate. So answer is option B and C. Then next one, which of the following bacteria is a non-heterocystous and non-symbiotic nitrogen fixing bacteria? So it is Acetobacter. So Anabaena, it is symbiotic and heterocystic, and Rhizobium, it is symbiotic and non-heterocystic. So E. coli is not a nitrogen fixer. So Acetobacter is the answer. Then next one, during electron transport, the extra energy carried by the electron is utilized in the formation of. So it is utilized in the formation of ATP. Answer is option A. Which of the following is not a secondary metabolite? So alkaloids, phenols, flavonoids, they are all secondary metabolites, whereas alpha ketoglutaric acid is not a secondary metabolite. So answer is option D. Then next one, number of ATP molecules required to fix one molecule of nitrogen is. So it is 16 ATP molecules are required for nitrogen fixation. Then hair cells are components of dash sensory organs. So hair cells are sensory cells of the auditory and vestibular system, and these are sensory cells of internal ear, essential for the senses of sound as well as in balance. So the answer here is option D, that is hearing. Then next one, match the hormones to the glands producing them. So oxytocin, it is by pituitary. Answer 1, 2. Then insulin by pancreas 2, 4. Calcitonin by thyroid and estrogen by ovary. So the answer here is option C. Then next one, myogenic heart contraction is initiated by. So the contraction, if you say that the contraction of the heart is myogenic, it means that signal for this cardiac compression arises from within the heart itself. That is that brain signaling is not involved here. That is called as myogenic heart or myogenic heart contraction. So answer is myocytes, and myocytes are cells of the heart. So the answer is option A. Then blood pressure is the pressure of blood on the walls of arteries. Answer is option A. Then which of the following nerve center is not involved in the regulation of excreting urine from urinary bladder? That is micturition. So it is dura mater. All the others are involved. Then which of the following organ regulates ionic balance in man? It is the kidneys. Answer is option B. Then the pituitary gland stimulates adrenal glands to make cortisol. Cortisol is increased during all of the following situation except. So cortisol levels are highest in the morning and lowest around midnight. Then as your body perceives stress, your adrenal glands make and release the hormone cortisol into your bloodstream, and its level can also be affected by illness also. So here the pituitary gland stimulates the adrenal gland to make cortisol, and cortisol is increased during all of the following situation except at night time. So answer is option D. The next one, which of the following is the functional unit of vertebrate excretory system? So it is the nephron. Answer is option D. The next one, a couple has a female child with the disease and 2 unaffected children. Neither parent nor any of the 4 biological grandparents of the affected child had this disease. Which one of the following is the most likely genetic explanation? So you can happily not consider X-linked dominant and X-linked recessive, because neither of the grandparents of the child are affected with the disease. So it is either autosomal recessive or autosomal dominant. And now if it was a dominant, then chances of other children also getting affected cannot be ignored. So the answer here is autosomal recessive. Answer is option A. Then which one of the following is correct for the linkage in Drosophila? So it is complete in males and incomplete in females. Answer is option C. Then next one, match the following definition and their nomenclature. Normal appearance of genetically controlled traits in the phenotype. So it is penetrance. Genes having more than one primary effect. Pleiotropy. Phenotype produced by environmental effect is same as the phenotype produced by a genotype. It is phenocopy. Both alleles of a gene express themselves in the heterozygotes. It is codominance. Answer is option A. The next one, which of the following combinations is the components of genetic variance? So the answer here is additive genes, additive genetic variance, dominance variance, and interaction variance. Answer is option B. We will see one by one. Additive genes, genetic variance is additive genetic variance. It comprises additive effect of the gene on the phenotype. Now suppose you have a plant with the allele A1 and it contributes 2 grams in weight, and then you have got another allele that is A2 and it contributes 4 grams. So for the phenotype for the genotype A1 it will be 2 plus 2, that is 4 grams. For genotype A1A2 it will be 2 plus 4, that is 6 grams. And for genotype A2 it will be 4 plus 4, that is 8 gram. Then the next one, that is dominance variation. So dominance genetic variation when some genes have a dominant component. That is in case the effect of an allele depends on the identity of the other allele at the locus. So if it is a dominant allele and a recessive allele being present, then dominant allele will be expressed. Then next one, interaction variance. That is genes at different loci interact in the same way as the alleles at the same locus interact. That is called as interaction variance. So genetic variance, it is its components are additive genetic variance plus dominant variance plus interaction variance. The next one, the unit of unit of distance in linkage map is centimorgan. Answer is option C. Then which of the following is the value of load score for 2-point mapping in human system? So load score or logarithm of the odds. It is a statistical estimate of the relative probability that two loci will be inherited together. They may and located close to each other, such as in the case of a disease associated gene and another sequence of interest. So in case of humans, the load score for 2-point mapping is +3 and -2. Answer is option A. Then which of the following chromosomal aberration in crossing or is a crossing over suppressor? So it is inversion. So during meiosis, homologous chromosomes are paired and sometimes an inverted region will prevent pairing for mechanical reason, and this will in turn prevent crossing over of DNA. Usually it is the paracentric inversion in which the inverted region doesn't involve the centromere, and they are called as crossover suppressors. So the answer here is option B. The next one, the gene is the basic unit of, so it is the basic unit of function, recombination and mutation. So the answer is option D. Then next one, match the following processes with their nomenclature. Genes are transferred by cell-cell contact, that is in the case of conjugation. A4. Genes are transferred inside virus particles, that is transduction. B2. Genes are transferred into cells as free molecules, that is transformation. C1. And bacterial cell gain access to foreign DNA through modified F factor, that is sex reduction. D3. So the answer here is option C. Then classification system for prokaryotic species based on the 16S rRNA was proposed by. It was proposed by Carl Woese. Answer is option A. Then next one. Which of the following is not a phylogenetic system of plant classification? So it is Bentham and Hooker system of classification. It is based on natural system of classification. So it is not a phylogenetic system of plant classification. Then in taxonomic hierarchy, the phylum was introduced by. So it was by Haeckel. Answer is option B. Then next one. Which one of the following organism belong to kingdom Protista? So it is slime molds. They belong to Protista. Sponges belong to Animalia. Mosses belong to Plantae, and blue-green algae, they belong to Monera. Then dash does not belong to gram-positive bacteria. So it is Azotobacter, it is gram-negative. Answer is option B. Then next one. Cell walls of prokaryotes are rigid and contain dash as main strengthening compound. So it is murein. Answer is option C. Then goblet cells are found interspersed in the dash epithelial tissue. So they are found scattered among the epithelial lining of organs such as intestinal and respiratory tract. And columnar epithelium covers the intestinal tract from the end of the esophagus to the beginning of the rectum. So answer here is option A, that is columnar. Then dash is an example of dead end phylum. It is Porifera. Answer is option A. Then which of the following is the state bird of Karnataka? It is Indian roller. Answer is option B. Then next one. Biodiversity rich Western Ghats traverse through how many states in India? So it is 6. That is Kerala, Tamil Nadu, Karnataka, Goa, Maharashtra and Gujarat. So answer is option C, that is 6. Then next one. In an ecosystem, the number of deer was counted as 2,500 in 1 year. In the following year, the number increased to 2,750. If the birth rate of the deer is 0.3, what will be the death rate? So the number of individuals in a population is 2,500. dn by dt is equal to 250. That is 2,750 minus 2,500. Now birth rate they have given as 0.3, that is n. And death rate we have to calculate. So growth rate is equal to dn by dt is equal to n minus dn. So 250 is equal to 0.3 into 2,500 minus d into 2,500. d into n. This is not dn, it is d into n. Okay. And this is b into n. The dn by dt, it is b into n, not dn, and it is b into n, that is birth rate into n, and d is death rate, that is d into n. So it is 0.3 into 2,500 minus d into 2,500. You will get approximately c, that is 0.2. So the answer here is option C. The next one. In an ecosystem, there was a large lake with plenty of fish. A human settlement grew in the vicinity of the lake and the fish population began to decline. Identify the possible sequence of events leading to the decline of the fish population. So if you see option B, lake polluted with sewage, algal bloom, that is eutrophication, it will result in eutrophication, it will result in decline in dissolved oxygen, and it will result in finally into increased fish death. So answer is option B. Then next one, a situation of ecological niche where a similar species can exist in the same area but use resources at different times is, it is resource partitioning. Answer is option B. Then next one, the artificial chromosome is used to clone DNA segment, is used to clone dash DNA segment. So yaks they use 200 to 2000 kb. So artificial chromosome is yeast artificial chromosome, and the answer is 200 kb and above. So answer is option D. Then dash is an area that acts as a boundary or a transition between two ecosystems. It is called as ecotone. Answer is option B. Ecocline is the gradual or continuous change of an ecosystem or community. That is, a wetland could be an example of an ecocline, in that it is gradual transition from terrestrial to the aquatic ecosystem. And ecological niche, it describes how a species interacts within an ecosystem. And edge effect are changes in population or community structures that occur at the boundary of two or more habitats. Then next one. Which of the following statement about case selected species is false? So case selected species are characterized by long gestation period lasting several months, slow maturation, and long lifespan and large body size. And they have extended parental care also. So which of the following statement is wrong is minimal parental care. It is extended parental care. Answer is option A. Fewer offspring. Yes. Large body size, yes, and long life expectancy. All that are correct except minimal parental care. It is extended parental care. Then next one. Miller's experiment on abiotic synthesis showed that simple molecule give rise to complex molecule. So simple answer is option C. Then FISH, fluorescence in situ hybridization, is used for the localization of dash on a chromosome. So fluorescence in situ hybridization is cytogenetic technique that allow localization of specific DNA sequences on the chromosome. So answer is option B, that is gene. Then next one. In a population, the frequencies of genes A and A have reached a steady state. Which of the following frequencies indicate that the population is in Hardy-Weinberg equilibrium? So if you add up all these, you should get 1. That is, if you add option A 0.064 plus 0.48 plus 0.09, you will get somewhere around 0.634. But if you see for option B 0.49 plus 0.42 plus 0.09, you will get 1. That is the answer. Whereas the others, option C is 0.36 plus 0.36 plus 0.09, that will be around 0.81, and D 0.49, 0.48, 0.36, that will be around 1.33. So that all these are not answers, but option B is the answer. The next one, a random change in the frequencies of alleles within a small population. So random change of alleles within small population is nothing but genetic drift. This question about genetic drift has been asked even in Maharashtra state eligibility test. So please refer to genetic drift before going to the examination. They are asking questions from genetic drift. I have seen in 2-3 papers now. Then next one. In evolutionary time scale, monkeys, apes and other mammals evolved during dash period. So it was during the tertiary period. So answer is option B. Then next one. Existence of 2 or more genotypes for a given trait in a population is called. So if you take pleiotropism, it means a single gene affects a number of traits in the same organism. Monotropism having only one form. Mutualism, it is symbiotic relationship, and polymorphism is it can exist in more than 2 forms. So existence of 2 or more genotypes for a given trait in a population is called as polymorphism. Answer is option D. Then next one. Female wasps have a choice of being a single foundress. She builds a nest and lays egg. Alternatively, she can join a colony of wasps and help in the colony activities, but cannot breed. Although there is no fitness advantage for the wasp, this behavior has evolved as an evolutionary stable strategy. Which of the following is probably one of the reasons for this? So the female wasp, it has the choice of being a single foundress. She will build a nest, lay eggs, and she can even join a colony of wasps and help them in colony activities, but cannot breed. So even though there is no fitness advantage for the wasp, this behavior has evolved as an evolutionarily stable strategy. So which of the following is probably one of the reason for this? It can be that she has a chance of taking over as the queen and then breed. So answer is option B. Then which of the following is responsible for evolution of multi-gene families? So a multi-gene family is a group of genes that have descended from a common ancestral genes, and therefore they have similar functions and similar DNA sequences. So duplication occurs primarily through uneven crossing over events in meiosis or of germ cells. So for evolution of multi-gene families, both duplication and unequal crossing over happens. Answer is option D. Then who proposed the great chain of being in the field of evolution? It was Aristotle. Answer is option C. Then which part of the brain is mainly involved in special memory? It is hippocampus. Answer is option B. Then of the several methods used for construction of phylogenetic tree, which one of the following uses character-based methods? It is maximum likelihood, whereas bootstrap and neighbor joining are distance-based methods. So the answer here is option C, that is maximum likelihood. Then read the following passage and answer the questions based on the passage. So you can read the passage on your own. Okay, so first question. How did researchers estimate the number of undiscovered species? So answer is option C, that is by constructing hierarchical tree-like structure where each individual species was classed in a series of progressively larger groups culminating at the kingdom. So answer is option C. The next one. The number of species on planet Earth have been estimated by different methods. Which of the following is not the total number of estimated species? So answer is option D, that is 7.8 million animal species. So this option, it is telling about only the total number of animal species and not the total number of estimated species. So which of the following is not the total number of estimated species, that is 7.8 million animal species. Then, what is the estimated number of mushrooms, molds and other fungi? It is option B, that is 611,000. Answer is option B. Then, dash percent of organisms have been entered in the central database, that is 14%. Answer is option A. So that's it. Please do like, share and subscribe. Thank you for watching my channel.